Can you see the first slide?
Yes.
Yep.
Okay,
Yep.
Excellent. So first of all, thank you so much to inviting me to this venue, and in particular to this one here for the 10 years anniversary. I still remember the very first conference, which was one of the most, well, delightful and most informative conferences I've ever been to in Barcelona. And I really appreciated that. So thanks very much for organizing this one. It's a remote one, but in any case, I have listened to two fantastic talks. Tony, thanks so much for a very general and fantastic overview and pointing or touching on some topics which are very interesting and important, which probably even include some political issues that we can discuss at a different stage. And then, of course, Barbara, that was amazing to see what you can learn from plants and other biological principles. Now, I'm a little bit reducing the complexity in a sense, only because I'm staying with insects. And Paul mentioned already, I started working in insects and then at some point thought it would be great to learn a little bit more beyond what the nervous system is doing to acquire sensory information, mainly through the visual system, and then turn the signal into control commands that could coordinate the flight engine here in a four-dimensional reconstruction down there of a blowfly, where ultimately just small little steering muscles are coordinating the maneuvers of a fly, or in this case here on the right-hand side, the micro-CT reconstruction that shows now the neck motor system, the muscles that are moving the head about when it comes to gaze stabilization behavior in flies. Now, what in the end is my interest here, and I share that interest with many people of you probably, is to understand how neuronal signals are actually used in some sort of control architectures to coordinate the behavior of the animal, and that's true for maneuverability of the fly or for stabilization reflexes altogether. And it's important to keep in mind that in these animals, one of the major features is, and that we will see a little bit later, that also causes or results in robustness, is that many sensory systems are employed, and that different sensory systems, like, for instance, the visual system, the compound eyes, are working in a different operating range. Here you can see on the y-axis the performance and gaze stabilization against the speed of a stimulus. This works at around 100 degrees per second, while the mechanosensory gyroscopic halter system is working at roughly 1 order of magnitude higher up and can respond to very fast stimuli. And of course, the question is, how are these different sensor systems combined so that ultimately the behavior is adequately controlled? Right, okay. Biology as a source of information to inspire novel approaches in engineering. Well, Paul and Barbara made both very good points. And I just happen to have this in my slide deck, where I make the distinction between 3 different flavors of bio inspiration or nature inspiration. I call one observational. This is often bio-inspired, nature-inspired, and that can be a little bit lean in a sense that it's basically using descriptive interpretation of biological phenomena, and then the attempt of trying to come up with some technology that may be inspired by that. The second one is what I would call bioprincipic. And in both talks before, we had that already. So it really is trying to understand the principles underlying function. And this is only possible to extract if we have a deep understanding of the biological design. And that's ideally from first principle. And then, of course, we have a mixture of both. But what's definitely true is that in any case, to identify and characterize biological design principles, we need to have an integrated approach that combines all the different methodologies we can get hold of from natural sciences and engineering. Well, here is one of the reasons maybe I can motivate why it's a good idea to look at flying insects. Now look at that left-hand side a video clip where you can actually see a blowfly in a roll maneuver just after taking off. And then on the right-hand side, you see a nice little hoverfly very nicely maneuvering in front of a landing site of some flowers. Now, the capabilities of biological systems are really unmatched by any man-made system. And this is true in terms of scale. So biology produces these things at very, very small scales. We learned about that before today already. Energy efficiency is a major point. Mechanical properties are unique in a way. And then the sensitivity of sensors is amazing. Obviously, the whole systems are totally autonomous and robust as Tony mentioned earlier. Now, these animals have had a little bit of time to come up with designs like this. And as Barbara mentioned, it's not really even that they are totally optimal, but they do exactly what they have to survive. And that's the criterion. So flies are like 240 million years old. That's when the first ones occurred. And that was even before any of the modern plants were present on Earth. And this allowed them basically to do something really important. They could learn to link sensors and actuators under the constraints that are given by their physical environment. Well, gravity is one of the time-invariant reference frames, if you want, that all animal locomotion coordination has to obey to. And that helps a lot. Of course, then in different situations and environments, it may have to require some adjustments, which animals can easily do by tuning their sensors, learning new links between motor action and sensory feedback, so forth. But they have had a long time to develop. Now, if we look at another maneuverable system that's man-made, this jet fighter here, that took maybe 20 years for R&D, and yet it will never, well, it's a fast development, but it will never, if we consider scale, reach the capabilities of any insects in the air. Now, I want to focus on visual processing here, and I brought a few videos to show you what some of the neurons in the nervous system of flies are capable of doing. Now, here in the center of this little screen, you can see a fly where you record the activity in an identified interneuron that is sensitive to directional motion, and I hope you responses as an alternating pattern of excitation and inhibition when you stimulate this particular neuron with motion in its receptive field. So you can do the same experiment at many different positions within the spherical receptive field of these animals, and then map for each of those positions what is the direction the animal responds best to when confronted with motion. And this is what you get. I call this a response field or a receptive field of a visual interneuron that is responding to wide field motion. And this is actually one of the major principles we can find in biology, and not only in the context of vision. In many cases, you have local sensors which would give you not exactly precise or unambiguous signals about what's going on in the world. But if you start integrating spatially across a huge number of those sensors, in this case, you would integrate about the output of motion detectors, which more locally analyze directional motion, then you can set up what you could call a matched filter that is extracting a very specific feature within the visual domain. Here, it is basically the motion pattern that occurs when the fly would, in this case, actually rotate around the vertical axis or go backwards in space. Well, in the end, what we learned over a long course of studies and so forth is that these matched filters for optic flow are likely to be designed in a specific way that is supporting the control of the natural modes of motion in these animals, which can be lateral modes or longitudinal modes that need to be controlled because they are potentially unstable. Here's another example for those matched filters where you have these cell types VS1, 6, and 8 responding to pitch motion here, roll motion, and an intermediate rotation axis in between pitch and roll. Oh, I should probably briefly explain what we are looking at is half a visual hemisphere. All the coordinates here are defined by elevation and azimuth. And so when the animal is rotating nose up around the transverse body axis, then the frontal visual field here at an azimuth of 0 degree, you would have maximum upward motion. If it's rolling, then you have maximum downward motion in the lateral visual field. So these cells are individual filters to extract properties that can then be used for motor control. Well, here is a very, very simple and in principle, a very efficient way of turning local sensor signals from elementary movement detectors into information that's presented to the motor systems exactly in the same sort of coordinate frame they need to control. So you can start from looking at the local analysis of motion using elementary movement detectors, and then you integrate them spatially to come up with a signal here that is already compliant with the coordinate system used by the motor system, in this case, to control head movements, where the blue axes here in the spherical representation of a motion axis are derived from the data we got from these interneurons analyzing optic flow, and the red axes in the same plot are the control axes of motor neurons driving the neck muscles. Right, so we can also do something that many of us like to do, and that's actually to see as a proof of concept study whether or not the idea we have about how the system is set up and it may function is actually working. In this case, oh, that's a little bit noisy. So in this case, it's in a linear control framework where basically the change of the state of the animal is causing an optic flow field in this visual environment. It is projected into these matched filters for optic flow, and then what comes out of that projection is a weighting matrix that is used to specify the control commands that are sent into the vehicle dynamics. So I saved you that video because the noise is a little bit interesting. But what I can say to explain what we see here in the video is a quadcopter that uses this principle and is negotiating through a corridor without colliding with the walls, controlling the forward speed and the bearing to make it through that maze. Right, so implementing biological design principles in robotic platforms, that's basically the first step for technology, and that's very clear. Now, to gain even a better understanding, you can then design something like biohybrid systems. And I have a very rudimentary explanation, and I really appreciated Tony's presentation, where he gave way more information on what the general definitions are. But from a very in a very broad sense, it's a connection between man-made components or systems and biological systems. And again, to get back to the point I made earlier, the major point that needs to be achieved for any biological system is using sensor information and use that information to control some actuators. And the way it's done is through the control mechanisms that link sensors to actuators. And this can be highly complex, or it can be very straightforward. And biology tends to the latter and tries to cut out all the supercomputers and all the massive computational efforts that are normally required in engineered systems. And it is done by, for instance, aligning sensory axes with motor axes and actually match them up so that there is no massive transformation between sensor signals and motor signals necessary. And this is possible in some cases because of something I mentioned earlier, because what ultimately happens when the actuators start any movement, be it locomotion or anything else, it is always within the context of the physical environment. And that has a couple of time invariant properties where really on the phylogenetic time scale, it makes sense to adapt to. Still, we have things like daylight cycles, as in day and night, and there are some adaptation mechanisms that are on a different time scale again, and most of the systems are capable of adjusting within limits to those changing conditions. And then obviously there are some situations where a fast adaptation mechanism needs to kick in, say for instance an insect flying from the open into the woods where there's less light and contrast available to control its motor action. Well, I don't go into more detail, but you will realize that in all these different examples, prosthetics, exoskeleton or robotic systems, or here biological sensors, sensory augmentation or sensory implants, here cochlear implant, you always have a technical component, but also the reference to the biological sensors themselves, and then the control architecture. This is going to be very interesting, in particular, over the last 10 to 15 years, artificial intelligence has kicked in and actually taken care of some sort of control principles or control architectures as well. Right. Now I have a few examples that are dealing with biohybrid systems that are not exactly focusing on vision, but rather on chemotactic behavior or olfactory behavior. And here on the left-hand side, this is a photograph of a crazy fly. That's a little quadcopter platform that's used at the University of Washington by Melanie Anderson in Tom Daniels' group to show that sometimes it's a good idea to just use a biological sensor to achieve certain functions. So what they really do in the course of a project that's funded through the Center of Excellence at the University of Washington that the Air Force is sponsoring, they use an antenna of a hawk moth, which we all know, and biologists most of all know, are using olfaction for finding mates big times. And they can just attach that antenna, you can see that here, held by forceps, to a circuit that's measuring the activity in the antenna, and that is then using, that information is then used to drive the quadcopter. We'll get back to that. And then, of course, in earlier venues of this conference, we have seen the silk moth system that was Ryohei Kansaki in the University of Tokyo has developed, where a silk moth with its antennae is actually detecting odor, and that influences the locomotion direction of the moth, rotating a ball. That movement is actually transduced into steering commands for the robot. So these are basically two examples where, in this case, the nose or the antenna is used to control a quadcopter. In that case, it is the whole animal as an olfactory sensor used to control a two-wheeled robot. I'll stay with the so-called smellicopter from the University of Washington because it's quite amazing what they could, in fact, do in their experiments. So what you see up here is the overall experimental arena. There is a odor source at this end here, roughly there. And we can see the drone doing a very typical behavior for hawk moths, right, actually, when they look for or try to follow odor plumes. When they have no odor plume available, they tend to do zigzag movements until they find the plume, then they go forward until they lose it again, and then go zigzag again until they get the track again. Now, that was the olfactory guidance, if you want, in that first run. In this one here, it is actually making the point that it also has a collision avoidance system implemented, which, though in this case is not a biological one, but basically it is using a laser rangefinder to make sure it's not colliding with any obstacles in the way. So this is really an interesting system, using olfactory information to steer a little robot and establish a biohybrid system that way. Now, we have done over the last couple of years some work on yet another biohybrid system, which hasn't got such a fancy name like a smellicopter, but we just called it fly-robot interface. And in this case, what we have is a fly as a sensor, if you want, that's mounted in the center of a two-wheeled robot. We can record the activity of directional selective neurons of the type I introduced to you earlier, and then use the signals of those neurons to control the movements of the robot. Now, why do we need this? And this is where it's really important to emphasize again that engineering helps neuroscientists to find new ways of investigating systems and trying to deepen the understanding of how sensory motor control in this case works. Because one of the major limitations, if you are operating on the nervous system and you want to understand how the nervous system and neuronal circuits work to coordinate behavior, then you normally have to fix the animal in place and it cannot move, and you have to control all the different all the different sensory modalities in a way so that you can make sense of the results. But in fact, the animal does not move in space. So you can then go for a very different system. That's the one here, where you use the fly robotic interface to study signal integration across different modalities and see how it all fits together when the animal is experiencing stimulation of their antennae by airflow or olfaction, we saw that earlier, or when the halteres, the gyroscopic sense organ, is experiencing angular velocities, and the visual system obviously is sensing optic flow. Now, here is the receptive field again of that neuron I showed you earlier, the H1 cell, and it has sensitivity to motion from back to front. Well, obviously, that's a little bit tricky to use those signals to control a robot if the robot is supposed to move forward. Now, an alternative or a way of getting out of that problem is to introduce an element of active sensing, which means the robot is not on a straight trajectory, but it's rotating left, right in an alternating pattern. And that means the excitation in this neuron is switching between increased activity and reduced activity, as you can see in this overall plot. So in the first plot here, you can see just the time interval during which the fly robot interface is turning to the left, which completely inhibits the activity in the neuron we are recording from. And then it turns to the right, which gives rise to the massive excitation in the neuron. This is just a zoomed in version that shows you individual action potentials, which can be separated and binarized in the system by onboard processing. And then it's converted into an instantaneous or continuous spike rate that is then used for the controls. Well, and the easy configuration we have it in at this point in time is just monitoring the spike rate, the instantaneous spike rate down here, and whenever that spike rate exceeds a particular threshold value, we trigger some sort of collision avoidance maneuver. This is a very old video we did a couple of years ago. So you can hear in the background the neuronal activity that is actually increasing whenever the fly robot interface is getting closer to the wall. and then it's rotating away. But this was an open loop experiment, not even including specific collision avoidance maneuvers. Now, we found through a couple of experiments that there's a very interesting dependence of the spike rate here. So here's again the recording on a parameter that is related to the proportion of rotation and translation in the movements. So for a high rotation component and a small translation component, which results in a turning radius of 5 centimeters here, you get a massive response. And the further you increase the turning radius, the smaller the response becomes. This is not the only dependence of the neuron in terms of stimulus parameters, but the other dependence of the response, which is plotted on the y-axis here, is on wall distance. And that, of course, is fantastic news, because that means in certain regimes for certain turning radii, you find in these dependencies a linear section, here again under closed loop conditions, I'll get back to that later, and under open loop conditions, where in fact you have a monotonous relationship between spike rate and distance. And of course, if you can exploit that, then you could use that as a control parameter to induce, even on a continuous basis, which we don't do as yet, a control signal that is making sure the robot is getting, staying off the wall. Interestingly enough, if you change the turning radius to very big turning radii, the relationship between the spike rate and the wall distance inverts. Well, that's a weird behavior in a way. We have some ideas of how this is brought about, but of course, what we normally do is we try to just deal with that problem by doing some model simulations, and that's what I did here. And what you can actually do is basically compare the plot on the left-hand side with the model simulation, which basically assume that we have elementary movement detectors with horizontal preferred directions, analyzing directional motion, and then integrating all the signals along the eye equator to result in an output activity of a simulated neuron. Well, what can I say? This is not exactly a quantitative match. I would rather say that we have some qualitative similarities, but we are far away from an exact explanation of the behavior or the spike rate of the neuron on the robot just based on motion detection. Well, you could say this is a little bit disappointing, but actually, if you remember, this was exactly our hypothesis, that the activity in the nervous system does not necessarily only depend on directional motion, but also on inputs from other sensory modalities, and this is sort of reflected or confirmed by this result. And we have independent studies done, or we did some independent studies showing basically that the Halter system, for instance, in the high dynamic range is contributing to the spike rate or increasing the spike rate of the H1 cell. And that is more or less exactly what we are looking for, the interaction between different sensory modalities that determine the spike rate and ultimately the behavior. Well, we did in a slightly more modern version of it, another experiment. There's no sound this time. Now, the way it works in this one here is, again, an oscillatory movement, but there's a bias of the fly-robot interface towards the wall. And whenever it gets too close and a threshold activity away and avoids a collision. And that's working really well. We can then do a kinematic analysis of what the robot is doing, in fact, and correlate that to the different, to the response parameters of the H1 cell and see what sort of relationships are important for us to improve on the control of that biohybrid system. One of the things we picked up in these experiments is that the spike rate is not only dependent on distance and on the turning radius. This is actually data taken with one turning radius, But if you keep the turning radius constant, then you still have another parameter, and that's the orientation of the fly robot interface relative to the wall. Right here, so this is another. Because it was facing a corner. And using this comparatively straightforward and simple mechanism of linking neuronal activity to the output of the robot or the maneuvers of the robot, it is still possible to negotiate those right-angled corners. Now, what's the next point, or what's also a possible explanation for discrepancies between pure visual information driving the rate in this neuron and in this H1 cell, and anything else that may contribute to its activity? And there was actually an incredible study a couple of years ago. When I stumbled across that study, I thought, okay, this is really one of the big findings, and I'll explain to you in a second why this is a big finding. The study was done in Drosophila, and we all agree, I suppose, that Drosophila is probably not the most, well, exciting and intelligent system on Earth, but, well, you never know. Anyway, so the experiments were done in the following configuration. The experimenters had arranged a fly so that it could fly in tethered mode, and they could measure the difference in wing beat amplitude, left and right wing, which is in this fly proportional to the torque the animal is trying to generate. At the same time, they recorded, and this is a feat in its own right, they recorded the activity of a neuron that's responding to horizontal motion. Key point in this experiment, though, is there was no patterned environment, so this neuron, whatever the animal would be doing, should not get any visual input, right? So this is, you would expect this, no visual input, the potential in the neuron should always stay the same. But if the fly is turning in a certain direction, which the experimenters can see by the difference of the wing beat amplitude, then you can measure in that neuron signals which basically anticipate and cancel out the predicted sensory feedback due to the own motor action. And this sort of efference copy basically is a massively important element in the control of animals to enable voluntary movements. So if it wouldn't be cancelled, the consequences of the animal's own movement, then it would never be able to do anything voluntarily, because it would always be switching on stabilization reflexes which pull the animal back into its original equilibrium state. So an efference copy or forward model can enable the animal to do voluntary movements, and it is absolutely essential. Now, what's also interesting about this is, well, we are talking about a prediction of the sensory feedback. And being able to do that, to develop forward models, predicting the consequences of your own action, is the necessary condition for any cognitive task solving. And you can observe that in a simple insect like Drosophila. And this is really remarkable. Now, and what we want to do, and I probably, because of the interest of time, I will have to skip this one, which basically gives you a general idea of how inner loop reflex or stabilization reflexes are linked to goal-direct behaviors, and how efference copies or forward models need to be integrated to enable voluntary behavior, and some of our work on how this could be implemented neuronally in local circuits, where we know what the different elements are likely to be, and that requires us actually to leave the safe terrain of time invariant linear systems and go to time variant transfer functions that can be adapted to the current environmental conditions. So what we then did is, in terms of the robot, because we want to have a system that we can use to investigate into this direction, we modified it to build robots where we can measure the wing beat amplitude, I'll get back to that later, and still have recordings from another platform from 2 H1 cells at the same time. Now this one here is very cute. You can see a fly now as viewed from below, where it's responding to pattern motion on the screen. You can see the differences in wing beat amplitude and analyze that as a function of the direction of the stimulus. Right, so here are some first tests. I might, in fact, skip this because I'm getting through. So this is using the wing beat analyzer, in fact, to avoid a collision with the bore. Oh no, now it's really stalling. Okay, so here is when the fly is changing the wing beat amplitude so we can steer away the robot. And this one here is a free fly run application of the neuronal signals. There's not much you can see here, unfortunately. I think the threshold was not set perfectly well. Anyway, so what are our next steps? So we want to record from 2 cells so that we can actually not just have wall following, but operate the robot in arbitrary environments. Then we want to have a combination where we monitor the wing beat amplitude differences and the neural activity, trying to learn in which way inference copies or forward models may play a role in the coordination. In studying the impact of different sensory modalities, I mentioned that very early on. And then obviously the ultimate goal is to operate the system in arbitrary environment, no matter whether what the contrast is, what the overall visual environment looks like. And then, of course, what one could imagine now at this stage is to think whether it would be good to just take the our knowledge now about the working of the motion vision pathway and have a fly actually flying a quadcopter. Right, thanks very much for listening. Here are the people who were involved in the project. Joseph, who has presented at the Living Machines conference a couple of times in the past. He is a postdoc in my lab. Jerry has worked on the wing beat analyzer, Jack Suttle as well, and together with Jin Wang and Yiling Wang was one of the first ones doing closed loop experiments with a platform, as was Yiran Y. Right, and then there were many little students who always worked on this project over the last couple of years. Thanks very much. And I give it back to you, Paul.
Okay, fantastic, Holger. Thank you very much. Very exciting new developments. So do we have questions? So Holger, then that you have these forward models kicking in now the control of perception. Can you give us a bit of a hint of the computations behind that? Do you see this as, let's say, wide field neurons that are playing a role here, or what's exactly going on? How complicated would these predictive models be?
Yeah. So unfortunately, there's, after the paper I referred to, the Kim et al. 2015 paper, there was only 1 or 2, there were more papers trying to trace back in the brain of Drosophila where these signals come from. It's not clear, but if you think about what needs to happen there, then it is probably a cumulative content of a neuron, say, that is correlating active motor commands with a sensory feedback as a result after a time difference of delta t that is required for the consequences to kick in. And you can imagine that as a very simple system where you basically do an online average of the consequences of your own motor action, and you update that with a weighting that is, say, dependent on the degree of your error signal or the difference between the current contents and the new one under different conditions. So in principle, there should be a very simple, straightforward mechanism in the nervous system enabling that. But of course, you know, it's never that easy in biology.
But would you think there's also an associative component required here?
Yes.
And then where is this associative component?
Well, I mean, this is, well, what we can assume is we have obviously always the possibility to take off the signals from the efferent pathways, which are driving the muscles or the controls, use that as one input to a neuron, say, and then use the current visual feedback that's present and combine that in a certain time frame. And that is some association. So there is some sort of associative learning component involved that requires a particular time window. And that's what we...
Exactly.
Yeah. So we did...
Why this resonates really well with the work we do on motor learning, where we have proposed this idea of counterfactual error. So you don't necessarily only learn to predict the outcomes of action, you also predict your expected error, and you use that to modulate your feedback controllers. Yeah. So this is definitely a discussion I would like to pursue with you in more detail, because that changes a little bit the game of let's say feed-forward control. Absolutely. I mean, well, I can tell you, I was so delighted when I saw that work, because you probably also know the old work by Van Holst and Mittelstädt, the reafference principle. And there was never really sufficient strong experimental evidence to really nail it down. There was some work in electric fish, of course. There was some work in the sensory context in crickets, auditory system. But now with this work in Drosophila, you can see the impact of forward models at the very optic flow processing. And that's amazing. Yeah. No, it is really astonishing, because these are minuscule brains, right? So absolutely, really exciting. Well, thanks a lot, Holger. Then I hope to see you again in the panel discussion. Other questions, let's go to Discord. Then now I thank you all very much, speakers and audience. This was a great opening session, 10 years of living machines. I want 10 more years of this. Thank you all very much.